It's my pleasure now to introduce the next speaker, Professor Jenny Graves, who you all know, an evolutionary geneticist who takes enormous delight in working with Australian animals, many of them, kangaroos, platypus, Tasmanian devils and dragons. Her purpose in doing this is to understand their evolution and compare it with our own, and to use that knowledge for many reasons. You can see everyone's CVs in your booklet, so I'm not going to go through them. I'm simply going to ask you to welcome Jenny to the podium so she'll have a little bit more time to talk. Thank you, Jenny.
Well, thank you, Suzanne, and this is a rather daunting task to survey Australian biology, past, present and future. I think when I look back that there's really three phases of biology research, really starting with organisms and observations of organisms, and then moving to cells and finally to genomes. And there's been revolution after revolution, not just in Australia, of course, but all over the world, in the way that people have investigated biology. I remember when I was a student, I think there were many things we couldn't do. We could do a lot of observation, but the techniques were simply not there to answer many of the questions we had. And over the last 30 or so years, I've watched in amazement as the technology has caught up to the questions, and I think even exceeded the questions, so that instead of poking around trying to get little bits of information here and there, we're now drowning in an avalanche of information. And I think really you can make you can look at many test cases, and what I've chosen to do is to concentrate on that most Australian of all biology, and that is looking at Australia's mammals and observing these changes in the kinds of biology that have been done in Australia. So essentially, first, I'll have a brief look at the way European naturalists discovered Australian biota, and then tell you something about the fundamental insights that we've been able to get into biology of all organisms, looking at Australian animals. And lastly, talk about what happens when Australia's unique fauna meets genomics. Well, of course, Europeans didn't discover Australian animals. They were well known to Indigenous populations 30, 40, maybe even 50,000 years ago from our cave paintings, and we also know that they had intimate knowledges of how these creatures ticked, because we have many bark paintings and a lot of Indigenous knowledge. But really, it was in the 16th, 17th, 18th century that Australians came face to face with very novel plants and animals of Australia. And Suzanne has already talked about Sir Joseph Banks, who is the naturalist on Cook's voyage, and there's a Banksia, there's many plants named after him, but you may not realise that he also commissioned one of his first famous drawings, paintings of a kangaroo, which was considered a really wondrous beast. So, in fact, even before this voyage, there were pictures going right back, maybe even to early Portuguese navigations, of these strange creatures that inhabited Australia. And strange stories abounded about the kangaroo, the Tasmanian tiger, the spotted cat, and that rather odd-looking creature down in the bottom left, which is a rather long-legged koala bear. And there were many stories about, well, how did kangaroos reproduce? Because one could see that they had a pouch, and in that pouch were little teeny-weeny kangaroos, and the thought was, well, they must actually grow out of the nipples in the pouch. And in 1790 or so, kangaroo was actually sent to London and greatly amazed the connoisseurs with the wonderful kangaroo from Botany Bay. And one creature was also sent to the king. People lined up for blocks to pay their shilling to have a peep at the wondrous Botany Bay kangaroo. Even more extraordinary were reports of an amphibious animal of the mole kind, which we now know to be a platypus. In 1799, a stuffed specimen was sent to the British Museum, where it was met with incredulity. In fact, they really believed that it was a fake, that clever Chinese taxidermists had actually sewn a duck's bill onto a beaver or some other mammal. And you can still see this specimen in the British Museum, and you can see where somebody has tried to find this with scissors, find where this was sewn on. But this was a real creature, and people then started observing it. And the big question was then, how did it reproduce? Because its internal reproductive organs looked more like a reptile than it did a mammal. But it was really a long time before a young Scottish naturalist came out and was able to find, with the help of Indigenous guides, platypus really caught in the act of laying an egg. And he sent this famous telegram, being Scottish and rather short of cash. It said, platypus oviparous, eggs meriblastic, which simply means, yes, the platypus does lay eggs, and these eggs really look rather like a reptile eggs. So again, there's enormous excitement in London and in Europe about these peculiar animals from Australia. Again, there are cartoons, there were poems like this one that appeared, and there's tremendous excitement about the strange habits of these strange animals. Well, that wasn't really science, it was more naturalism and observation. Really, the early science would have started in something like 1892, when very young professor of anatomy in the University of Sydney gathered around him, again, very young fraternity of Duck Malloy. And I'm indebted to Houghton Obisco for alerting me to some of these marvellous characters. And they were specifically looking at Australian animals with the idea of testing the new ideas of evolution that Charles Darwin had put forward. And this was a wonderful period in which this group of scientists, and their work is still around today. One of these young people was Peter Hill, and the Hill collection is still very famous. They looked at the anatomy and the physiology and the reproduction, particularly of these animals. And this was a marvellous period, but it really ended in 1906, when these people went back to England with their knowledge, and there was really rather a dark patch when the new professor of the medical school thought, well, we have no business to be looking at Australian animals, we're medical sciences here. And again, there was really very little interest and no funding for Australian research until Harry Waring arrived in Western Australia, and there was a renaissance led by him and by Houghton Obisco and Jeff Sharman, who are fellows of our academy. And they're really the fathers of what we know today as research on our own native animals. I would have to say, though, that the funding has not improved for work on Australian animals, but I think what I want to tell you now is how very important they've been in understanding some fundamentals of biology. So there's a phase of looking at the anatomy of marsupials and the physiology of marsupials in the 50s and 60s, and it was found that these animals are just remarkable inside as they are outside. And for instance, here's a picture of the internal organs, reproductive organs of a kangaroo. In fact, the kangaroo has 3 vaginas and 2 uteruses. And the reason that they don't fuse as they do in placental mammals is that the ureters from the kidney go the wrong way round. And Hugh has always told me that this was a real mistake of evolution because it meant that the babies had to be very, very tiny and they couldn't be large as they are in eutherians. It was also discovered that, at least in some of the animals, the penis is double. So really, I imagine these animals can double their fun. So marsupial development turns out to be an amazing collection of differences. There's a very fundamental difference in the structure of the egg and the embryo and the way it develops, so you don't get a mass of cells inside as you do in a mouse and all other placental mammals. So the structure is much more primitive than that. But the really amazing thing is that the young are born very, very early, after just 20-odd days inside. They're born, and even without any back legs or any gonads, they crawl up into the pouch of the mother and attach themselves to a teat, where they stay for some months as they develop. And so this is an absolutely marvellous model for development, and Marilyn Renfree has exploited this dramatically to look at the stages of development, at very young development of a mammal. Even better than that, you're able to feed what is essentially a fetus, feed drugs or hormones, because you can put a little tube into its mouth. So this is an amazing model for development. And it's too bad, I think, that the medical fraternity has never really recognised that marsupials offer this tremendous model. Kangaroo milk is amazing because it changes dramatically in its composition during the life from a very tiny little being to a furry being that hops in and out of its mother's pouch. The amount of carbohydrates go down and lipids and proteins go up. And again, this milk may be full of important growth factors, which we're just starting to find out about. Well, Australian mammals meet genomics quite recently, but probably you all know that the revolution in molecular biology started in the 50s and 60s with the discovery of the structure of DNA. And it's only now that we're able to start looking at the code embedded in DNA. Yeah, as you probably all know, DNA is a very tiny, thin little molecule, but very long. We each have about 2 meters of DNA in every cell of our body. But it's not just like a bowl of spaghetti. It's complex with proteins in a very specific way and contracted into the bodies that we can actually see that we call chromosomes. Now, marsupials have absolutely wonderful chromosomes. They're famous for their chromosomes. They're enormous chromosomes, and there aren't very many of them. But probably what you may not know is it's essentially the same genome that we have. So there's very few differences in the number of genes or the type of genes or what they do. It's simply the packaging. But because they're so beautiful, and I have to say they're beautiful enough, they actually made a stamp out of kangaroo chromosomes in 2003, that a lot of very basic work on chromosomes has been done with marsupial chromosomes because they're so easy to observe and manipulate. One thing that has been able to be done, and a lot of this work has been done in my lab, is to tag the molecules of DNA from different chromosomes. You can actually sort chromosomes out and you can tag it with different fluorochromes, and then you can lay it on the chromosomes of another species and look at the homologies. So, for instance, these are 3 chromosomes from one species of kangaroos labelled in different colours, and applied to the chromosomes of a different species that have bigger chromosomes, and you can see that essentially it's the same old chromosomes, but they're just jammed together. And I think this came as a big surprise how very simple a lot of these evolutionary rearrangements were. In marsupials, essentially you can trace the same blocks of chromosomes through all marsupials in the world. And so we've been able to use these sorts of data to construct exactly how these blocks of DNA were moved around in the last 100 or so million years. So this is quite a paradigm of chromosome evolution, and it's really only now that the placental people are being able to use the same sorts of techniques, because it's a lot more complicated. Also, when you look at the structure of marsupial chromosomes, there's many things that you can do with marsupial chromosomes that you can't do with human or mouse chromosomes. One thing is these mysterious parts of the chromosome called the centromeres, which is how chromosomes are pulled around in division. They're very big and complicated in mouse and human, but they're very small and quite tractable in marsupials. And so you can actually pull the DNA out and find out what sequences are in it. It's also possible to look at a family of jumping genes, and extraordinarily, these actually jump around, and up to the present, they're not dead jumping genes at all. And we've been able to look at what happens in situations, for instance, interspecies hybrids will activate these. And the ends of chromosomes are also very specialised. Our own Elizabeth Blackburn got a Nobel Prize for figuring out how these ends are maintained. A certain group of marsupials, starting with the Tasmanian devil, turns out to have most extraordinary telomeres that somehow are rather imprinted in male and female meiosis, so they come into the egg either big or little. And here you can see the two sets, the two genomes, one has bigs and the other has littles, and this seems to be a completely new way of controlling the ends of chromosomes. Well, I'm sure you've all heard of the Human Genome Project, the idea to map all the genes and sequences in the DNA of a human. It took 10 years, it cost $3 billion. And that was in 2001. If you look at other placental mammals, by 2003, mouse had also been done, and a couple of birds, and a couple of fish and a frog. By 2007, there were lots and lots more placental mammals, and a couple more birds and reptiles, but you can see there's a great big gap in the phylogeny between 100 million years, which is when placental mammals radiated, and the last common ancestor we shared with reptiles and birds. And we thought at the time, well, it would be great to have something to fill that in, and we do, and that's where Australian mammals really have it all over other animals, because they're more distantly related to humans than are, say, elephants and cats and dogs. And so that was a very powerful reason to sequence some of our own animals. So I was asked in 2003 to write a proposal to sequence an Australian animal, and of course I had to choose a kangaroo. They came back and said, well, actually this is American money and we think we should sequence an American marsupial, so that's why they did the opossum. But there were 24 of my Australian colleagues were authors on that paper. Fortunately, there is no American platypus, so they spent $40 million sequencing the platypus shortly after. And in collaboration with Marilyn Renfree's group and many other groups, we were finally able to do the kangaroo a couple of years ago. Now, almost every week, I get requests to, please send a bandicoot liver by the next mail so we can sequence that as well. And there's now data on the Tasmanian devil genome, and there's a koala genome project going on right now. So this gap is going to be well and truly filled, and we're going to be drowning in information pretty soon. My particular interest has been in a part of the genome which is very, very weird, and that's our sex chromosomes. As you all know, I'm sure, in humans, as in other mammals, the female's got two X's and the male has got a single X and a Y chromosome. They're both rather peculiar. There's about 665 genes on the X, but they do many things, not just sex. But the Y is very strange. There are only 45 genes on it, and they largely have male functions. One of these genes we know is very important. It's the sex gene. We know that if you have a Y chromosome, you're male, and that's it, and it doesn't matter how many X chromosomes you have. So there must be something on the Y chromosome, and this something was suggested to be a gene called ZFY. And that's really where marsupials came into the picture, because we were able to show that that's the wrong gene, because in marsupials it's actually not on the Y chromosome. There's another gene called SRY that my student Andrew Sinclair went to London, and it was he who led the successful hunt for that gene. So that's where marsupials really made it into genomics, because they did things somewhat differently. Well, sex chromosome story is a really interesting story. The sex chromosomes of a kangaroo are much like sex chromosomes in a human. Those little chromosomes at the end of the sex chromosomes in a kangaroo. But it's a little bit different, because individuals that have two X chromosomes and a Y, which in humans would be male, is actually intersex. It has a penis and a pouch. And so that gives us quite a different idea of how these sex genes evolved. So we know from our studies with both kangaroos and elephants and humans that the sex chromosomes evolved from a perfectly ordinary pair of chromosomes. One partner acquired a male-determining gene. That was a kiss of death for that chromosome, because other genes then took on a male advantage role, and to keep these genes together, recombination with the X was suppressed. All sorts of dreadful things happened to the Y then by deletion and mutation, and the Y chromosome slowly degrades and degrades, and this is pretty much what it's like in marsupials these days. But it can even be lost, and in some groups of rodents it is actually gone and replaced by something else. So we can calculate that the Y chromosome will be gone completely in about 5 million years, and that's why I call it a wimpy Y chromosome. So marsupials and also monotremes have been extremely important in trying to figure out where our sex chromosomes came from and what they do. So we know that marsupials have X and Y pretty much like humans do, but reptiles and birds have all kinds of other sex chromosomes that are not related at all. And it turns out that monotremes, when we looked at platypus, have 10 sex chromosomes, but they're completely unrelated to humans and mice and kangaroos. So what this means is our sex chromosomes are very young, surprisingly young, only about 150 million years old. The invention of our sex-determining gene SRY is fairly recent. And again, this was a big surprise to everyone, and marsupials and monotremes did it. The other thing that has been well known in marsupials is an epigenetic change. Now, epigenetics, as was explained 2 days ago, there's a lot of information on top of the DNA sequence of the genome that tells genes when, where and when to be expressed. And the sex chromosomes, X chromosome, is a famous example of this, because females have 2, males have only 1, and that's of course not fair. So what happens is that one X chromosome in females is genetically inactive. And when we looked in marsupials, it turns out that's also the case in marsupials. Here's 2 different ways of looking at for the inactive X chromosomes. It's there. I concluded, well, it's just like humans. It's not at all. In fact, it turns out that this chromosome is always from the father. And that was actually the first indication of what we now know as genomic imprinting in a mammal. So everybody will tell you it was discovered in 1990. No, this was 1970, and we've known all about it for a long time. And in work that was spearheaded by Marilyn Renfree and some Japanese collaborators, we've been able to look at the first steps of how this imprinting has been set up in marsupials. And cancer, you've already heard from Kathy Belov yesterday that the Tasmanian devil is heavily endangered because of a horrible facial tumour disease that turns out to be actually the cells from an original founder animal that got a cancer of a Schwann cell. So this cell can be transmitted from animal to animal by biting. And so it's been possible to use the same genomic techniques to look at not only the origin of the cell, and it expresses genes that are typical of a Schwann cell, but also what's happened to the chromosomes. And you can see the chromosome 1 here has been totally blown to smithereens, and probably one of the genes on chromosome 1 might well be a driver for cancer. We've also been able to look for, surprisingly, human genes by going far away into marsupials. And so we keep on accidentally discovering human genes. Just my little lab has discovered 14 human genes, all by accident because we're looking for something and something else turns up that's not supposed to be there. That's not very helpful for getting grants because you can't get a grant by saying, I aim to clone 14 human genes all by accident. That doesn't work. Equally important is finding that there's some genes that are supposed to be very important actually not there. It also has been very instrumental in looking at how a particular family of genes, like, for instance, the globin genes, evolved using genomic data from kangaroos and platypuses. Well, of course, evolution is really where Australian animals have all the answers. The platypus particularly is most amazing animal. Was many discussions, well, is it really a mammal? Well, it has fur and it makes milk for its young, but there's no teats. And besides, it lays eggs like a reptile and it makes venom. And one of the reviews we wrote, somebody drew this cartoon where a duck was mated with a beaver and turns into a platypus, which we thought was very funny, particularly since there's a Dreamtime story in Aboriginal Dreamtime story, that's exactly how the platypus came to be. So not the least is the amazing sex chromosomes of platypus. It was found that there is no homology at all with the genes on the sex chromosomes in humans and kangaroos, but the surprising thing was there was homology to the bird sex chromosomes. So the telegram that we were going to send on Roger Short's advice from a Bowden conference was, platypus is furry bird. Well, what about the future and where does Australian animal fit into it? Well, you've heard my doomsday prediction that we're going to run out of a Y chromosome in 4.6 million years, and we do need men because we can't reproduce without the sperm. There it goes. I don't think we're going to have to wait that long for extinction because I think we're obviously trashing the environment, and the next 100 years could be mass extinctions. We could see a lot more bushfires and a lot of severe droughts in this country. The world population is now 7.1 billion years, and Professor Walker on Tuesday reminded us the fact that there's really only enough water and energy for 7 billion people. So what's this going to mean for Australia? If there are severe droughts, then that means we're going to get severe land erosion. A lot of our soil ends up in New Zealand because we insist on having hoofed animals that disturb the fragile soil in the centre. Perhaps we should consider getting rid of our hoofed animals and eating kangaroos. Is genomics going to save us? Well, genomics is getting way cheaper. Here's a line showing that to begin with, it was something like $3 billion to sequence the human genome, $40 million to sequence a platypus genome. You can do it in about $3,000 today. So genomics is getting a lot cheaper, and there's a body I belong to called Genome 10K, which aims to sequence every vertebrate. So I think it's not crazy to think that it will be extremely easy to sequence all the Australian mammals that anybody has an interest in, and not only sequence the DNA, but also the epigenome, looking at the modifications of the genome that turn genes on and off. So I think there's going to be immense amounts of information that is going to come tumbling out over the next few years that we might be able to use. Or how can we use it? Well, first of all, who needs a model mammal? We're always being told that we should all work on mice because mice are the model mammal. But now we have genomes of Tasmanian devils and dragons. We can certainly use these when biology throws up a situation that's worth exploring. What about antibiotics? We have trashed our antibiotics by feeding them to animals. We need new ones. You probably remember from Kathy Velov's talk yesterday that kangaroo milk might be a source of potent new antibiotics. The skin of the pouch makes antibiotics that protect the vulnerable marsupial young. So it's not crazy to think that we might have a new source of antibiotics here. Platypus venom has got some remarkable peptides in it that cause excruciating pain, and we might be able to learn something about pain medication from that. So there's all kinds of strange situations in nature that we're now able to explore using genomics. And lastly, well, what about conservation? Australia has a terrible record of extinctions of mammals. There's something like 18 Australian mammals that have become extinct over the last 100 years or so. Can we use genetics to monitor populations and even to manage populations? And one thing that is being done now is to look at contraception in overabundant animals like koala and kangaroo islands. And this is work that Marilyn has been doing for many years. It's also going to be possible to monitor health and to maybe find out the cause of problems like the devil facial tumour disease and figure out what to do about that. There's also a lot of mammal pests like the rabbit and the fox, and it might be possible to find out ways in which we could control those pests. And finally, is it possible to resurrect some of the species that we have shot to death, like the Tasmanian tiger? This certainly isn't possible at the moment, although it's going to be perfectly possible to sequence the tiger from all the skins and all the museums, but actually putting it together into a sensible chromosome and particularly putting it together in a sensible cell is at the moment quite beyond our reach. But I hope that this will not be permanently beyond our reach. So my conclusions are that we have an amazing fauna in Australia, and we really haven't, we've used this to find out all sorts of things about the biology of all animals, but it's not been a well-funded venture, and perhaps now it's possible to get sequenced. It will be much more recognised that we can get answers about all fundamental problems from our own fauna. But I think we do have unique opportunities with the genomic age because we don't need models anymore, and there may be new products and new opportunities that we can now exploit. And I'll just put one of my favourite Fireside cartoons up there. So don't laugh at weird mammals. There's a little echidna wandering past some dinosaurs, which I think is very funny. Thank you.
I'm sure you have some questions for Jenny. I think that was a brilliant overview of how our knowledge about our own creatures has advanced over the years. Yes, over here.
Oh, sorry. Is it on? Yeah. Thank you for this wonderful talk. It just reminded me of a paper I read from, it is François Jacob, who published in 1977 a paper in Science entitled Evolution and Tinkering. It really shows that not only at the molecular level, but in Australia now with this Australian, you have really that evolution has been working like a tinkerer, putting different parts together, and when it just happens that situation arises, then you have you can end up with so-called strange animals, which are wonderful creatures that you like wanted. If you would like to comment on that yourself, please.
Well, I couldn't agree more. I think Australia is a wonderful place to study evolution because we have been isolated for a long time, and that means our plants and animals have found different solutions to the same sorts of problems. It amuses me very much that Francois Jacob once said that what is true of E. coli will be true of the elephant. And I think we now find out that that's not right at all. We still have a long way to go to investigate elephants and kangaroos, although we know quite a bit about E. coli.
Over here, Francis.
Oh, thank you, Jenny. I enjoyed that. The platypus shows bird-like connections. What about the echidna? Does that show relationship to perhaps reptiles?
Echidna is fairly distant from platypus, but in fact it shows almost the same genome. There are just very few rearrangements. Essentially, chromosomes have got jammed together a bit like they have in kangaroos. So again, their sex chromosomes are related not to other mammals, but to those of birds.
Roger Short, Melbourne Uni. Jenny, superb talk as always. But can I just raise one terminological inexactitude?
You would.
It is calling marsupials not placental mammals. Every single marsupial depends on having a placenta. So could this meeting please get away from this sort of European terminology that marsupials are somewhat lower mammals, they're not even placental.
I'm sure Jenny would be happy
Well,
about that.
of course, the problem is, as you well know, because we've been arguing about this for 20 years, the alternative is metatheria, which means not quite true beasts. So placentals are eutheria, that means true beasts, but marsupials again are denigrated. So I think I'll leave it to my betters, Hugh and you, to please argue this out, just make something up. But marsupials is a bad term too, because many marsupials do not have pouches, and marsupium, of course, means pouch. So there is really no good answer.
Okay, I think we better be moving on. I have to say that I'm really excited about the possible new antibiotics that might come from this field, because we're in desperate need of new antibiotics.